Okay, this video is in regards to week 9, and we're talking about the structural and functional plasticity of the muscle. The truth is that all the slides until slide 17 that starts talking about the molecular motor of the muscle is basically background and basic knowledge that you should know by now. So I don't want to lengthen the video, so go over it. And if you have any questions, just refer them to me. So jumping to slide number 18, the molecular motor, also known as the native molecule, is the hexametric complex of 6 proteins. So I think that the use of the word motor is very interesting because it's the origin of force and power for instance, a car, if you think about the word motor. So I also think that this is the right word to use in regards to the muscle. I think that the major takeaway point from the presentation today is the many differences in isoforms of the heavy and light chains of the muscle and how diverse muscles actually are. We are taught to believe that there's 3 types of fibers. That is very inaccurate, and I would like you to focus on that watching the video and going over the presentation, making sure that you understand how many different actual types that do influence performance there are in muscles. So when we look at the myosin heavy chain, the MHC, so to speak, we're talking about 3 general parts that we see in the chain. We have the rod, we have the subfragment, and we have the globular head. When you look at these 3 parts, I want you to think about a hand that can grab something. Okay, so there's the rod, which is the arm. There's the globular head, which is that functions like a hand. All right, and then there's the subfragment that connects between the two and transitions energy and all the processes that lead to the function of the myosin. Okay. The rod is actually an enhancement of the structure of the packing of the chain itself, okay, and causes it to look like a thick filament. All right, so it gives it its structure. When we're talking about the head itself, that is the part that actually binds to actin and generates force to move the actin back and forth relatively to the heavy chain. When you look at the head, it has 3 parts as well. We have the catalytic domain, meaning that is where energy is going to be, or ATP is going to be broken down, hydrolyzed. We have the converter domain, meaning the part where energy or chemical energy is converted to mechanical work, and then we have the arm / lever that actually moves and creates movement within the muscle. Let's see, so we talked about that. We can go on to slide 20. So when we're talking about different isoforms, we're talking about mutations or alleles, different forms of the same chain. And when you look at familial hypertrophy or cardiomyopathy, for instance, changes in these chains play a role in those conditions and disease. When you multiply the amount of heavy chains, isoforms, but those of the light chain isoforms, you get multiple options and diversity within the population. So we can no longer stay at the level that there are 3 types of fibers. Yes, that is a very general division of the fiber types, and actually if we were to analyze each and every option that is consisted by those isoforms of the light and heavy chains, we'd have way more muscle types. The speed of the muscles, their ability to produce power and work are definitely a direct result of the combination of the chains that you're going to find in the sarcomere. The heavy chains are by far the more dominant factor that will influence the force-velocity relationship. Just remember that there's 2 relationships and not only this one, which is this relationship of the force-velocity relationship is a major factor in the work of a muscle, a skeletal muscle. So we are accustomed to type 1, type 2A, type 2B, and type 2X muscle fibers. The type 2B is actually not really represented in humans. And we know that we have different abilities to every and characteristics to every fiber type. We also have other isoforms that we see throughout the development of the human body. There is a type 2L and a type 2M. One is specific that are more specific to certain areas of the body. For instance, the larynx and the mastication accordingly. So the type 2L is called that way because it's in regards to the location of the larynx and so on and so forth. And you can see, for instance, that the type 2M, which is unlikely to be found in skeletal muscles, is super fast, meaning, completely anaerobic. We also see that in early periods of life, this is on slide 22, we see more embryonic isoforms that appear, meaning that they have some kind of advantage or developmental stage that is relevant to embryos, but do not exist in the adult person or even after birth. We can see that we also have cardiac isoforms that include the alpha and the beta isoforms, with the beta being the slower cardiac isoform, meaning the more aerobic one. When we talk about isoforms of light change, myosin light change, we also have many isoforms that have been identified, number 1, number 2, number 3, and even within those isoforms we have sub-isoforms that have been identified. Some are fast, some are slow. When we look at the second isoform of the light chain, we see that those dominate mostly smooth muscles. So if we now go farther into the structure and function of the muscles of the sarcomere, now we have to take into consideration isoforms of all the sarcomeric proteins, meaning topomyosin, tropomyosin, and so on and so forth. And how you need to multiply those isoforms by all of the other isoforms, and you can understand that there are multiple uh, um, outcomes possible to a skeletal muscle. We kind of went over the stages of sarcomere work. So on slide 24 and 25, you have information about that, but you've basically learned this information before. Okay, let's jump to slide 27. When we look at the work of a skeletal muscle, we identify in general 4 processes. We have excitation, we have coupling, contraction, and then relaxation. I just want to say, to be accurate, that relaxation is the lack of contraction. Now this may seem very trivial to you, but there is no neural way to induce relaxation rather than the lack of contraction. So this is very, very important. There's no way that you're sending a neural signal telling the muscle not to contract, what we call to relax. So once again, relaxation is simply a period where the muscle is not contracted. The way to do that is to utilize chemical barriers that prevent the option of the muscle being excited or coupled or contracted. One of them, which is the easiest one that we have already learned about, is removal of calcium from the equation. So this is where the SR comes to play with the batches or content of calcium that it has. Okay. Since we talked about this, I'll kind of refresh your memory. In general, we have an impulse. The calcium is kept in certain areas, stored away with a membrane. That membrane or that storage of calcium is canceled. The calcium kind of disperses into the sarcomere and connects to actin. Actin is then rotated around its longitudinal axis, and that allows the globular head of myosin to identify that and catch or connect to the actin when paid by ATP, or when ATP is hydrolyzed and the energy is absorbed. And then that movement, the relative movement between the two filaments, can be achieved. Upon the will to stop contracting the muscle, we are taking away calcium. Calcium is reabsorbed, and we have a repolarization of the SR. So the speed that all this can happen depends on the isoforms of the pump, which is related to the isoform of the heavy and light chains of the sarcomere. Let's go over the mitochondria. Once again, a lot of this is basic knowledge, so I'm just going to jump forward. Obviously, the mitochondria has a major and center role to play in any cell. The mitochondria has also a negative aspect to it, such as the production of ROSs and initiating apoptosis, which is the the death of the cell. Obviously, when we are exercising someone, the demand for ATP produced by the mitochondria goes up. We know why. We know that aerobic ATP production is preferred over anaerobic under certain circumstances. And we know that ATP is also created by the transfer of electrons as pure energy. We know that the Krebs cycle is the supplier, so to speak, of hydrogen to the electron transport chain. We know that the electron transport chain plays with levels of energy to basically operate an enzymatic complex called ATPase that connects ADP with P to create ATP. That's probably the simplest version of the story. We know that we have 4 complexes within the ETC that together, step by step, move electrons between ions. We know that the ATP is charged with those electrons. And as I said in the past, ATP is the molecule, but it serves in a way like a credit card that you can charge with pure money, and those would be the electrons. Let's see what else. Okay. Okay. When we look at the mitochondria, we see that some are within the myofibrils and some are in between. This structure obviously optimizes in certain conditions and physiological situations the shortest distance from a mitochondria to a needing chain. Let's go on to slide number 33. And okay, slide number 33 is kind of basic, so I suggest that you go over it. All right, going a bit into the muscle isoforms, which is the major aspect of what we're going to talk today. We need to understand that when we look at a muscle fiber, we want to identify both the mechanical and physiological properties. In this case, the physiological properties can be called also chemical properties in a way. At the end of the day, what we want to know about a muscle is how much power can it produce and how fast or slow it's fatigued. So basically we're talking about the rate of power, some kind of aspect of strength produced per time. Okay. At the same level, we can look at energetic efficiency, which we always care about as physiologists, and calculate the amount of energy needed in order to produce a certain amount of strength. This is where obviously all the isoforms come in, the dominant metabolic pathways that are dominated by the enzymes, the aspects of anaerobic versus aerobic production of energy, and so on and so forth. When we want to see what are the major origins of the characteristics or different characteristics of muscle fibers, I think that we can break it down to the contractile filaments or proteins, calcium, the amount of calcium cycling that is going on, the SR itself, and the mitochondria that it has. Let's continue to slide number 36. When we think about structure and function, which is a major principle that we always talk about, then you can see in slide number 36 how the structure, meaning are the sarcomeres structured parallel or in series, and we see that according to the type of the myosine, meaning the heavy chain, the ability to function changes. So if the sarcomeres are in a certain muscle fiber are structured in parallel, this is the best way to generate maximal force. If they are structured or arranged in series, we can see that we find maximal shortening velocity or excursion. Usually we say that the cross-sectional area of a muscle is one of the best indicators of the ability to produce force. This has to be, of course, normalized to cross-sectional area. But when we talk about shortening velocity, not the total outcome, rather than the dynamics of the muscle itself, then definitely the heavy chain isoforms are going to affect that and give us a different dynamic of work. In the muscle, in a way, chemically, and also because it's a biophysical weight, in the sarcomere, obviously the ability to control the rate of release of calcium and its reabsorbent is a major factor that contributes to the ability to produce force. And part of that cycle, especially the later one, depends on enzymes. Okay, this ability to release calcium and reabsorb it is completely dominated by and governed by the SR. Means that the last point on slide 37 is extremely important. The rate of work is basically SR dependent. Remember that. The rate of work of a muscle is SR dependent. The SR is about 2 to 6% of the cell's volume, with type 2 having the largest SR volume, meaning the 6%. We have isoforms of SRs that have been identified as well, and they are accommodating, so to speak, the type of muscle needed, so we can see a difference between those in cardiac cells and those in skeletal muscles. We can see that we have RYR1 and RYR2 that are subgroups of the RYRS, but we can also see that the RYR2 is commonly found in all tissue types. So the RYR1 is more unique than the two. We can see that when extracellular calcium is absent, the RYR1 subtype becomes a major factor in excitation, contraction, and coupling processes. In the fast fiber, we'll find those more at about a ratio of 10 to 1. Let's see. Interpreting that information means that the RYR1 subform is basically adequate for higher velocity force production than the 2. Let's see. We see on in slide 40, we're talking about the enzymes SERCA1 and SERCA2A that are highly efficient. They definitely are influencers of the rate of work production or power production, and we also see that the volume density of the SR, how dense is the SR, is one of the factors that have been indicated as a main influencer of the ability to stop the contraction of a muscle, what is called here the relaxation rate. When we look at mitochondria volume or size, we see that in the slower fibers, aka the aerobic fibers or the red fibers, the mitochondria tend to be in average up to 3 times more than in white fibers or fast fibers. We can also see, of course, different key enzymes, which makes sense because if a fiber is dominantly aerobic, it will need aerobic enzymes versus anaerobic. Polymorphism. We haven't gone through genetics yet, but polymorphism is the idea of different forms of gene expression, meaning different forms or isoforms of proteins with certain function in the body. So as stated here in slide 42, we're talking about high resolution understanding of multiple isoforms at different levels of all the components of the sarcomere, and that's the idea that is behind slide 42. Talking about the muscle fiber pattern, if you remember, we talked about a division of 50-50 in the population, meaning that the population in general has no specific direction genetically, meaning that most people will not have unusual anaerobic abilities or unusual aerobic abilities. Right now, at least, evolution is directing us to the middle, meaning that we want, so to speak, to eat the cake and keep it all as a whole. We are not betting on anaerobic abilities to survive or anaerobic. And remember, when we talk about function, it has to align with the needs of surviving in nature. When we talk about adaptation, we can also expect to a certain extent different rates or extents of adaptation according to the overall combination of all the isoforms of all the components of the sarcomere. This, of course, complicates things greatly when we talk about exercise prescription and adaptation. I'm guessing that somewhere in the future, maybe, I don't know, 100 years, we would be able to analyze relatively easier exactly what complex combination of isoforms someone has and prescribe exercise according to that. it might even be identified without the need of any tissue example biopsy, rather than simply analyzing the genes. That's where it may be going. When we talk about transitioning of muscle fiber types from one to the other, there is a scheme that is suggested by Pete and Sterum, and you can see that if adaptation, if change between muscle fibers is going to happen, there are certain options, but not all exist. So we can see that we can go between type 1 and type 2A. We can go between type 2X and type 2A. And we can go between type 2X and type 2B. This scheme is not always followed, meaning that in theory these options all exist, but for some reason they are not happening. What we see in general is that a anaerobic muscle fiber, what we call a white muscle fiber, might become what we call a pink one, meaning increased oxidative ability. That's as much as we usually see, and once again there has to be a good reason for that to happen. We must understand that although we love to think about adaptation, changing the content and the structure of a muscle, which is a major organ within the body related to movement, which is related to survival, is not an easy decision. And that's why you won't see it happen that easily, and definitely the extent of change is not going to be crazy big. Let's talk about plasticity. We are on slide 41. So talking about plasticity, we need to talk about the content of the organ or of the tissue, and of course the cells. You can see that, or you know that when we look at a cell, there are components of the cell that contribute to its stiffness, and there are components that contribute to its fluidity, and there are components that contribute to its plasticity, and there may be components here and there that contribute to its elasticity. So when we look at the estimated average muscle fiber, it has about 20,000 myonuclei. The extent of the coordination between these mechanisms of coordination is unknown. So we do not understand or do not know yet to what extent there is coordination between the nuclei, but we know that there's a certain amount of it or rate of it. There is evidence and an ever-growing body of knowledge that certain muscle fibers have local expression, gene expression at the level of mRNA, which is interesting, meaning that it may be a leading mechanism that allows adaptation at the fiber level. Very interesting idea, and definitely can lead in the future, to genetic engineering of certain muscles with certain abilities to improve rapidly due to exercise. It also means that certain aspects of adaptation to rehab / exercise are very localized at the fiber level, not the whole muscle tissue and not the whole DNA tissue. level. So this is very interesting. It also means that the adaptation or transition from one muscle fiber type to the other might be simply the accumulation of very localized changes rather than something that goes through the whole muscle. At the genetic level, this is very important and different, two different dynamics of changing something. Okay, slide 48, we are addressing the idea of structure and function, which is a principle that I think that we've talked about a lot. And some of the things have already been mentioned in prior presentations, but I'm happy to talk about them or mention them again, at least in the presentation. We've talked obviously about the muscle being an organ, a tool that converts chemical energy, in this case ATP, into mechanical energy, in this case movement. We know about the muscle's ability to produce force during shortening and lengthening, and even isometric work is dependent on 4 factors. The length-tension relationship, the rate of activation, which would be the neural aspect of muscular work, the force-velocity relationship, which we've learned about, and the rate and degree of relaxation. Once again, I'm using relaxation because you'll see it in other places, but actually we're talking about the rate and degree of the lack of activation. When we are looking at these relationships, they are directly influenced by the content and structure of the muscle, as we said. So that's the first point on slide 50. The question asked is the process one directional or two? Meaning, does gene expression affect the muscle only, or can changes in the function of the muscle and the stimuli to the muscle affect gene expression? You have at the last point of slide 50 the 3 components of the muscle, which is basic knowledge, so I think you should go over that. And this is basic anatomy. Okay, when we look at the CE component of the muscle, it is accountable for both the length tension and the force-velocity relationship, meaning that it's a major component in the structure function of the sarcomere. We see that the sac represents elastic elements, and the pec includes the elastic elements that are parallel. And these 3 components are part of what we call the Hill model, with the researcher Hill that suggested this module and proved it. Is this module perfect? No. It doesn't necessarily explain the muscle's performance or behavior during lengthening contractions, but it does cover the majority of options for the muscle. At a personal note, once again, since there are so many isoforms and changes, I believe that there should be multiple modules of activation and performance and behavior of the muscle. I don't think we can get away with such a simple model, but it is a good place to start with. Okay, we'll stop here and continue in part 2. Stay tuned. Enjoy the video.